So we just in our notes had talked about proteins, amino acids. Proteins are extremely important, as we talked about, because they make up our body's tissues, our muscles, our skin. They are make hormones and antibodies, and also this group of proteins called enzymes. Enzymes are really important, and so we're going to talk about them in some detail here. So enzymes are basically a protein that speeds up a chemical reaction in a living thing. Basically, we stay alive because of millions of small chemical reactions going on in our cells and our bodies all the time. And our body needs these special proteins called enzymes to allow all of those chemical reactions to happen. So the term for that is a catalyst. A catalyst is a molecule that speeds up a chemical reaction. And enzymes are organic catalysts. They're proteins that do that. They speed up these chemical reactions. Basically, they allow a chemical reaction to take place when they lower the amount of energy it needs for these to happen. And enzymes are very, very specific. A single enzyme only helps a single chemical reaction to take place. So if we have thousands of different chemical reactions taking place in our body, we have thousands of different enzymes allowing them all to happen. And the key thing to remember about enzymes and about all types of proteins, it's their shape that is extremely important. They have a complicated shape, and that complicated shape allows them to sort of connect with other molecules and cause these reactions to take place. Now, we usually simplify this. Like, this is a pretty simplified example. Really, an enzyme isn't a nice little sort of jigsaw puzzle shape. It's super complicated. But we can simplify it and use these sort of symbols. So here we have this green thing. It represents an enzyme. It's a protein. And here we have a molecule that this enzyme is going to work on. Okay. And so what happens is the shape of the enzyme kind of matches with the shape of whatever molecule it's going to work on. And so when they're together, they kind of connect. And we see that here. The enzyme is binded to this substrate molecule. And then what often happens is the enzyme changes slightly and it causes something to happen. In this case, what did this enzyme do to this purple molecule? So if we go from this to this, what's happened to it? It's broken apart. What's the term we just used for that? Jordan, what was the term you answered? Digested. This molecule has now been digested. It's been broken into two smaller pieces, and this enzyme helped it to happen. Now, maybe this would have broken up on its own, but with the enzyme connecting it all and allowing it to happen, it happened much more quickly. So this is an example of an enzyme that digested this molecule. Lots of times, enzymes are named with the ending ASE. So for example, an enzyme that takes sucrose, just table sugar, and breaks it up into its two components, that enzyme is called sucrase. It's just named the same thing, but it ends in ASE. And so here's another example, another enzyme. Oftentimes, the enzymes need some helper molecules. For example, this enzyme, which affects this substrate molecule, on its own, this substrate wouldn't fit properly. But once this helper molecule comes in, That makes it so the shapes are going to match again. Lots of vitamins that we need in our diet. You might take, if you take a vitamin each day, lots of those substances in those vitamins act as these coenzymes to help enzymes work. So enzymes are these big molecules with very specific shapes. Now let me show you this again. Has the enzyme changed at all after it helped this reaction take place? Does it look like it's changed? No, it's basically the same. The enzyme can then be used again. It's not destroyed in the process. So after this enzyme helps digest this, it can connect to another of these purple molecules, another substrate, and do it again, and do it again. These enzymes don't get used up. They kind of get reused many times. The place where the enzyme and the substrate are going to connect has a term. It's called the active site. Makes sense. The site where they're kind of interacting with each other is called the active site. And the molecule the enzyme is working with is called the substrate. So the enzyme and the substrate, and then what's released are called the products. So again, here's another digestion. Right? This substrate connected with this enzyme at the active site caused a change to happen, broke this substrate into two pieces, and now the enzyme can work again. Now this enzyme did something different. What did this enzyme do? What's that? Yeah, it's called synthesis. It's the opposite of digestion. This enzyme allowed these two smaller molecules to all connect, and then a change happened to these molecules, and they were combined into one. So this enzyme took small molecules and built a larger molecule. Enzymes can do many different things. These are sort of two common examples, digestion and synthesis. And so this enzyme, when it's done, again, it's not changed really. It can be reused again to do this over again. Now, it used to be thought of that the enzyme and the substrate molecules all like fit together like a puzzle. Scientists have since learned it's not that simple. That the shapes don't always match perfectly. In the 1950s, they found out that actually once the substrate molecules start to attach, often the enzyme changes to mold to them. So it's not as much like a puzzle piece or a lock and key, but the idea is the same. It is the shape of all of these molecules that's important. And so here's some examples. Here are some digestive enzymes that are found in the human body. In our stomach, we have cells that produce this enzyme called protease. And this enzyme, its job is to break up proteins. And it produces amino acids. It's digested proteins into their smaller components. In our small intestine, we have another enzyme called sucrase. It takes sucrose, again, and breaks it up into simpler components, monosaccharides. In our stomachs, we have lipase, which takes fats and breaks them up. In our mouth, we have an enzyme called, our saliva has this enzyme called amylase. It takes starches and breaks them up. So these are all examples of digestive enzymes that do this process. Now, how well these enzymes work, how quickly they work, depends on different factors, like temperature. So, here's a graph. These graphs you're going to see a lot. This graph shows the temperature and how well the enzyme's working. So typically what we find is as enzymes are heated up, they start to work faster because molecules are moving faster, they're more likely to interact with each other, and so the enzyme activity increases until a certain point. And then what happens after that point is if it gets too hot, the enzyme stops working as well. We can see that line, it drops off. What happens as these enzymes get heated up, their shape can start to change. And if the enzyme shape changes, it no longer fits in with the substrate and it can't work. So in the human body, most of the enzymes we have work best at body temperature, at 37 degrees Celsius, 98.6 Fahrenheit. And this is why, one of the reasons why having a high fever can be dangerous. Because if our fever gets too high, if our body temperature starts to get up to 103, 104, 105, we're getting into temperatures that could be fatal. Because the enzymes in our body, they're getting past the point when they work properly. And the chemical reactions in our body can start shutting down. And we can't survive that. So that's why temperature is really important. Body temperature has to be within a relatively narrow range. pH. So pH is a measurement of acidity, how acidic something is. And enzymes work best at certain pHs. We could see that in this graph. Pepsin, which is an enzyme found in our stomach, it works best at a low pH. You can see that in this graph. Salivary amylase in our saliva works best at a neutral pH, around 6 or 7. Alkaline phosphatase works best at a higher pH. It's in our small intestine. And so if the pH changes too much one way or the other, again, the enzyme's not going to work as well. Also, increasing the amount of the substrate can increase the rate of the chemical reaction to a certain point, but not unendingly, because eventually the enzymes are saturated. They can't handle any more of the substrate, and so the graph levels off. So as you can probably guess, the pH of our stomach, we'll talk about this eventually, our stomach is very acidic. And therefore it makes sense that the enzymes that's in our stomach works best at an acidic pH. In our mouth is pretty neutral. And so the enzymes in our mouth work best at a neutral pH. Through natural selection, our enzymes are sort of adapted to working best where they're found in our body. Oh, just the last thing, pH, because we just mentioned it. Let's talk about that. pH, it's a measurement. It's like if you can measure the temperature of water. You can measure the amount of oxygen in the water. You can also measure the pH. And pH is a measurement of acidity. It's a scale. The pH scale goes from 0 to 14. 7 right in the middle is considered neutral. Like if you have pure water, it has a pH of 7. It's neutral. It's not acidic at all, and it's not basic at all. Here's a graph of different substances. So some things are slightly acidic, like milk is a little bit acidic. It has a pH of 6.5. Coffee is pretty acidic. It has a pH of 5. So we get down to the low end, things like that are dangerous. Battery acid has a pH of 0.5. Can burn your skin. That's how acidic it is. At the other end, things that are basic, things like cleaners, soaps, bleach, seawater, for example. These are things that are at the other end of the acidity. They're called basic. And so, like we said, different enzymes work best at different places on this pH scale. Okay, let's stop there.